Biological oxidation is a fundamental process in biochemistry. It involves the removal of electrons or hydrogen atoms from molecules, typically facilitated by enzymes. These reactions, known as redox reactions, are always coupled. When one molecule is oxidized by losing electrons, another must be reduced by accepting those electrons. A key concept in biological oxidation is the electron transport chain. This is a series of protein complexes located in the inner mitochondrial membrane. These complexes transfer electrons from donors like NADH and FADH2 to a final acceptor, usually oxygen. This electron transfer creates a proton gradient across the membrane. The main electron donors in this process are NADH and FADH2, while the final electron acceptor is typically oxygen, which combines with protons to form water. This proton gradient is used in the process called oxidative phosphorylation. As protons flow back into the mitochondrial matrix through ATP synthase, ADP is converted to ATP, the primary energy currency of the cell. The enzyme ATP synthase acts as a molecular motor, using the energy from the proton flow to catalyze the synthesis of ATP from ADP and inorganic phosphate. Enzymes called oxidoreductases play vital roles in catalyzing oxidation and reduction reactions. These include oxidases, which use oxygen as a hydrogen acceptor, dehydrogenases, which remove hydrogen from substrates, hydroperoxidases, and oxygenases. For example, malate dehydrogenase is an enzyme that catalyzes the oxidation of malate to oxaloacetate in the citric acid cycle, with NAD+ serving as the electron acceptor. Coenzymes play a crucial role in biological oxidation. Molecules like NAD+ and FAD act as oxidizing agents. Flavin adenine dinucleotide, or FAD, consists of a riboflavin attached to an ADP molecule. During oxidation reactions, FAD accepts two hydrogen atoms to become FADH2. The principles of biological oxidation are also evident in photosynthesis, where redox reactions occur in chloroplasts rather than mitochondria. In photosynthesis, light energy powers the movement of electrons through an electron transport chain, ultimately leading to the reduction of carbon dioxide to form glucose. Recent research in biological oxidation emphasizes the role of molecular redox switches in governing oxidative stress responses. Particularly important is the dynamic role of cysteine amino acids in proteins, which can undergo reversible oxidation and reduction. This has opened up the field of redox proteomics, which studies how protein function is regulated through oxidation and reduction of specific amino acids. Recent studies have highlighted that RNA is more vulnerable to oxidative damage than DNA. This RNA damage has been linked to several serious diseases, including neurodegenerative conditions, atherosclerosis, type 2 diabetes, and various forms of cancer. Transition metal catalysts have gained significant interest in the field of biological oxidation. These catalysts improve the efficiency and selectivity of oxidation reactions while reducing environmental impact and energy requirements. Let's explore some key examples of biological oxidation processes. In photosynthesis, the water-splitting catalyst in photosystem II oxidizes water to produce oxygen, protons, and electrons, a fundamental process for energy production in plants. The cytochrome P450 system is crucial for metabolizing drugs, toxins, and other foreign compounds by adding oxygen to their chemical structures. Interestingly, intestinal microflora produce gases resembling the ancient Earth's atmosphere, suggesting an evolutionary adaptation to anaerobic conditions. Measuring porphyrin levels can identify biochemical damage and exposure to toxins like arsenic and mercury, serving as useful biomarkers of oxidative stress. Current trends in biological oxidation research are focusing on several interconnected areas. A systems biology approach examines oxidation within complex biological networks, while redox signaling research explores how these reactions regulate cellular pathways. Therapeutic applications target oxidative stress for new disease treatments, and the field of enzymomics is advancing with improved experimental assay systems. Researchers are expanding the redox proteome by studying the role of specific amino acids, while also investigating antioxidant defense mechanisms against oxidative damage. Antioxidant defense mechanisms protect cells against oxidative damage. These can be divided into enzymatic and non-enzymatic defenses. Enzymatic defenses include superoxide dismutase, catalase, glutathione peroxidase, thyrodoxin reductase, and peroxyredoxins. Non-enzymatic antioxidants include small molecules like glutathione, vitamin C, vitamin E, carotenoids, and flavonoids. A key finding in recent research is that antioxidant enzymes play a more significant role in defense against oxidative stress than small molecule antioxidant compounds.